Can you guys hear me all right? I know it's very, very loud in here with my 3 attendees. To those of you watching on vodcast, shame on you. I mean, really, come on, you can't support this? Who's going to laugh at my jokes? Lots of awesome jokes. All right, so I passed out this sheet for this section of the book. Whoops. I don't know what I just pressed. This is the Chapter 9 study sheet. It's listed on the website that you guys have a link for somewhere. That's 9A, 9B, and 9C. It's all 3 sheets, so it's 3 pages. All Chapter 9. We'll be going through that today. All right. So any questions from anything prior? Very excited to be here, enthusiastic about biochemistry. Yeah, that's what I'm talking about. All right, so we'll go through this pretty quickly. You've got everything in front of you, so like I've been doing, hopefully you have the chance to watch this again if you don't need it. First couple pages of Chapter 9, the special stuff, overview. We're going to be dealing with amino acids that are the precursors for a bunch of different stuff. So amino acids is a precursor for neurotransmitters, for thyroid hormones, for the nitrogenous bases, ATGC and AUGC, and all of that stuff. So this chapter is all about a bunch of different synthesis pathways. So there's like 8 or 9 different pathways we're going to go over. Just small pathways, short pathways. Page 98 and 99 are overviews of kind of what we're going to be covering right here. Talks about the heme group, the creatine group, and the purines and pyrimidines, which we're going to talk about. Different hormones, melanin, some neurotransmitters, etc., etc. We're going to start right on the top of page 100 because that's where it gets important. Now, very importantly, right up here at the top, glycine. So what you need to know in this chapter is you need to know the rate-limiting enzymes, just like every other pathway. You need to know the substrates and the other enzymes, but most importantly, you need to know what amino acids contribute to what. So, for instance, this is your first little hint right here. Glycine contributes both the carbon and the nitrogen, not really important. You need to know that glycine is a part of hemes, creatines, and purines. Rule of 3 is that's a great test question. Which of these is glycine not a part of? Glycine is a part of hemes, creatines, and purine bases. Often the distractor is pyrimidine bases because we're going to talk about the 2 different nitrogenous bases. Some are purines, they're larger, and some are pyrimidines, which are slightly smaller. So glycine is a part of hemes, creatines, and purines, not the pyrimidines. So as we get into this, you're going to see that I'm going to point out the amino acids that go into each of these. I'm going to point out that glycine is an integral part of creatine, and we're going to show you what other stuff is used in each of these, and you got to memorize those for sure. So talking down here about heme structure, some general stuff, I would know that it is comprised of a porphyrin ring. It's the ring structure we're going to assemble when we go through the synthesis pathway. It's a porphyrin ring. The porphyrin ring is made of 4 pyrrole rings joined together, TQ. Heme is composed of a porphyrin ring. It's made of 4 pyrrole rings joined together. It doesn't say it here, but it shows it. Oh, here you go. Surrounding the iron. Very importantly, the central ion in a heme group is iron. That's what gives it its oxygen binding capacity, because that is the purpose of a heme group. It's part of hemoglobin, and hemoglobin is used in what? In blood, to transport oxygen. So when we're synthesizing hemes, it's with the goal of attaching it to a globin, which is a protein chain. It's a soluble protein chain because it's a globin as opposed to, remember the fibrous stuff we studied. So we're going to synthesize this heme structure by building 4 pyrrole rings, cramming them together, making a porphyrin ring, and then putting an iron ion right in the middle. With that iron ion in the middle, we have a heme group. That heme group can be attached to a globin and distributed, etc. So we need to know where we make heme groups. Where's the tissue that synthesizes? What is the tissue that synthesizes everything? The liver. So liver is one of the places. What else is going to synthesize red blood cells? What do we call those types of tissues? Erythroid. Good. So down here it says heme synthesis occurs in erythroid cells, and erythroid is just anything that produces an erythroid cell. Erythroid-poietin or whatever is anything that produces an erythroid cell. And an erythroid cell is a red blood cell. So anything that produces red blood cells is going to be able to do heme, as well as the liver, because the liver does everything, as our rule normally implies. You guys following along so far? Cool. So Now we get down to the pathway, and I'm going to ignore this. On the back of the first sheet, at the very top, it should say heme biosynthesis. It's kind of blotted out a little bit. This is your shortcut for heme biosynthesis. Heme biosynthesis goes from right here to right here. Now you notice below where my thumb is, it says catabolism. So after we talk about the synthesis of hemoglobin, we're going to talk about the breakdown of hemoglobin. So heme synthesis occurs at the top. What was that amino acid that heme is used in hemes? What was that amino acid that glycine? Where else is glycine used? Hemes, creatines, and purine bases. Hemes, creatines, and purines. They all kind of rhyme, right? Purines, hemes, creatines, all go with glycine. Okay, so we start out heme biosynthesis where? Erythroid cells in the liver. I even told you that on here. It starts out with the combination of glycine and succinyl-CoA. Glycine and succinyl-CoA come together and they form the first substrate of heme biosynthesis, which is delta aminolevulinic acid. Delta aminolevulinic acid. I'm going to stumble over that all night, I promise you. Delta aminolevulinic acid, aka delta ALA. Delta ALA. That is the first true substrate of heme biosynthesis. That reaction, succinyl-CoA plus glycine going to delta aminolevulinic acid, is performed by delta ALA synthase. Surprise, surprise, right? It's a synthase enzyme that is making what it's telling you it's making. Why can't we name it for the substrates? Normally we name our enzymes for the substrates. Why can't we name this one for the substrates? What? True. But we can't name it for the substrates because there's more than one substrate, right? So we can't call it succinyl-CoA something or glycine something because there's two of them. So we have to name it for the products. The product is delta ALA, and what's it doing? It's delta ALA synthasing. Delta ALA synthase is the rate-limiting enzyme of all of heme biosynthesis. The rate-limiting enzyme of all of heme biosynthesis. Do you think he's going to ask about it? So what amino acid is important for heme biosynthesis? And what TCA intermediate is important for heme biosynthesis? Succinyl-CoA. So make sure you know that succinyl-CoA is involved in the TCA cycle, which means that if he asks you something about a tricarboxylic acid or something like that, that deals with the TCA cycle that you know, and he's referencing heme biosynthesis, that you know he's talking about succinyl-CoA, which is one of your starting substrates. Again, both of them to go through delta ALA synthase. Now, notice that over here it says mitochondria. That means that this part occurs in the mitochondria. Heme biosynthesis occurs in both the mitochondrial matrix and the cytosol. The mitochondrial matrix and the cytosol. That's a TQ if I ever heard one. Heme biosynthesis occurs in two subcellular locations, the mitochondrial matrix and the cytosol. So, delta ALA is made in the mitochondrial matrix by delta ALA synthase. That reaction is a decarboxylation reaction. We lose a CO2. All decarboxylation reactions happen through PLP. Anyone remember what that stands for? PLP. Pyridoxal phosphate, which is aka vitamin B6. Now I'm going to start really drilling you guys on these because that blue sheet that I gave to you, I'll put it up on the monitor for those playing the home game. This blue sheet, bluish, is the Biochem 2 vitamin chart. You will also find it on that website. This goes with Chapter 11 and Chapter 12, which is water-soluble and fat-soluble vitamins, both of which are on this upcoming test, which means that you're going to know these vitamins by the time you get to the next test, which means that all of the stuff that's testable material on this test that involves these vitamins is very important for you to know. So if you wanted to pull an example reaction, you need to know that vitamin B6, aka pyridoxal phosphate, is the decarboxylator. And that's something you're going to have to memorize in Chapter 11. Vitamin B6 does all decarboxylation and all transamination reactions. We've talked about that before. What are our big three transaminations? Pyruvate to alanine. Bam. Give me one. They rhyme. Oxaloacetate to aspartate, and glutamate alpha ketoglutarate to glutamate. So your big three, pyruvate to alanine, oxaloacetate to aspartate, and alpha ketoglutarate to glutamate. Pyridoxal phosphate does decarboxylation and transamination. Anytime you see a decarboxylation or transamination, it's this guy. Now there is a different type of decarboxylation. We'll talk about that in chapter 11. So, rate-limiting enzyme of heme biosynthesis is delta-ALA synthase. We've delta-ALA synthase, we've made delta-ALA. Delta-ALA has been transported out into the cytosol, where it is combined with another delta-ALA that was made by delta-ALA synthase in the mitochondria. We have two out in the cytosol, and they are delta-ALA dehydratase. Delta-ALA dehydratase, producing porphobilinogen. Now, the AKA for porphobilinogen is very important. It's a pyrrole ring. The AKA for porphobilinogen is a pyrrole ring. I think I have it written in there right there for you. How many pyrrole rings compose a heme? How many pyrrole rings compose one heme? 4 pyrrole rings. How many delta-ALAs are in 4 pyrrole rings? How many delta-ALAs are in 4 pyrrole rings? 8, right? Because we take 2 delta-ALAs to make 1 pyrrole ring, and we have 4 pyrrole rings. I don't think he's going to test you on that. I just want to make sure you guys are paying attention, and you can do simple math since we're at a doctoral program, you know. So, it's fine. Delta-ALA synthase combines succinyl-CoA glycine. We make 2 delta-ALAs. We ship them out into the cytosol. We've changed subcellular locations. 2 delta-ALAs condense. Dehydratase, what's it getting rid of? Dehydratase. What's this enzyme taking out? Not hydrogen. It's dehydrating it. What do you do when you dehydrate something? You take out water. Fantastic. So that's a common way we make bonds. Remember, that's how we make protein bonds, is we dehydrate that. We don't call that dehydration. We call it condensation, but it's the same thing. Water's leaving. So this combines 2 delta-ALAs by stripping some oxygens and some hydrogens, making water and combining them at the site. Please note, there are 2 delta-ALA enzymes. You got to know that synthase is the rate-limiting enzyme. It's not dehydratase. Don't get confused. I have a little note over here. This is one of the TQs for the chapter. This enzyme, delta-ALA dehydratase, is inactivated by lead poisoning. Inactivated by lead poisoning. One of the clinical correlates. So we've made our pyrrole ring. Now, how many pyrrole rings do we need for one heme? 4. So we're going to take 3 more pyrrole rings. We're going to go through a bunch of different intermediates that you don't have to know for the test. I'll point out how many you get to skip on the next page, and you'll rejoice in joy. You need to know 3 enzymes and 4 intermediates for this. Porphobilinogen, pyrrole ring. Combine 4 of those guys together, and we get protoporphyrin 9. Protoporphyrin 9 is the last intermediate, the last substrate before it becomes a heme. Because what's it missing? Missing iron. So protoporphyrin 9 is the 4 pyrrole rings joined together. It's the 4-ring structure. The last thing we have to do is we have to add an iron, which means we need an iron. So we take our iron, and we combine it and make a heme group. Now, ferrochelatase is the enzyme. Is that one going to be hard for you guys to remember? No. Why? This ferro, which means fantastic. Chelation is what they do when they attach something to something else. Like you can chelate stuff in the blood. It's a way of getting rid of heavy metals and toxins. You can chelate stuff out of a substance. It's what they use in anticoagulation tubes. They chelate all the calcium out so the blood won't coagulate, et cetera, et cetera. So chelation is a method of combining stuff. So it's combining an iron molecule with a protoporphyrin, which is 4 pyrrole rings, which is 8 delta-ALAs, which was made by the rate-limiting enzyme, delta-ALA synthase, to make a heme group. Now you can add a protein, specifically a globin chain, and you can make hemoglobin, myoglobin, or cytochromes. So where do we see cytochromes? Cytochrome B, cytochrome C1, cytochrome A, A3, electron transport. Which, guess what? What do they do in electron transport? Transport electrons. So these guys are good at carrying oxygens, they're good at carrying electrons, which means clinical correlate, if you have a decrease in delta-ALA synthase, you're going to have trouble not only with oxygen transport, but with electron transport. Don't know if that'll come up, but that's where the cytochromes are involved. So we've made heme. Any question on that part so far? Okay, that's our first pathway. We'll skim through it on here. I'm not going to go real slow on here because we talked about it all. Condensation of glycine and succinyl-CoA forming delta-ALA. Delta-aminolevulinic acid synthase is the rate-limiting enzyme, uses B6 or pyridoxal phosphate, which does decarboxylation and transamination. Chapter 11, you're going to have to know that. Rate-limiting enzyme. Shows all that fancy stuff up at the top of the page up here, succinyl-CoA and glycine, delta-ALA. 2-delta-ALA out in the cytosol now, combined to form a porphobilinogen, which is a pyrrole ring. Diffused to the cytosol, condensed to form protoporphobilinogen, delta-ALA amino dehydratase, inactivated by lead poisoning, clinical correlate, and then lots of other stuff until finally you get down here. Iron is incorporated into the protoporphyrin ring, catalyzed by ferrochelatase to form a heme molecule. Now here is the full pathway. You need to know these substrates, these enzymes, and these substrates and enzymes. Not even this one that I highlighted. It says meh, because I highlighted the wrong one. Just these guys right here. You get to skip all that. Is that exciting? He's never, to my knowledge, tested on that. Boards doesn't test on that. So, not important. It's part of heme biosynthesis. It's there for your reading delight and all your free time. All right. We get that. Questions on heme biosynthesis. What amino acid does it use? Glycine. Fantastic. How many pyrrole rings make a heme? 4. How many iron molecules make a heme? What's the enzyme that adds the iron? Ferrochelatase. What is the rate-limiting enzyme? Delta-ALA synthase. Fantastic. Good job. What's up, man? So, now we're going to move on to heme breakdown. You guys cool with that? No questions? Heme breakdown comes right after heme synthesis on the little sheet I gave you. It's all in the book, but we're going to skip through my sheet so you guys know what's going on, and then we'll move on from there. So, first thing, we're considering that we actually have hemoglobin right here. Pretend that says hemoglobin, not just heme. Because the first thing I do is I break off the globin chain. So this only happens with hemoglobin when it actually has the globin chain attached to it. The globin chain is amino acids or a protein, which can be broken down into individual amino acids. When it's broken down, it also releases free iron, which goes back into iron stores or goes anywhere we need it to so we can use it later. And it decarbon monoxidates. I don't even know what you call that. But it ditches a carbon monoxide right here. The breakdown of heme breaks down into two things. Heme itself breaks down into biliverdin, and carbon monoxide. Don't think that'll come up, but just in case. First breakdown product of heme is biliverdin, and it gives off carbon monoxide. Biliverdin is converted to bilirubin. This all happens in the blood, and biliverdin is given off in the blood, is converted to bilirubin, and bilirubin is then transported to the liver. So what this is right here happens in the liver. Actually, I'm sorry. This happens in the liver. Those two steps right there. Bilirubin is very lipid soluble. It's very non-water soluble, which means it's transported to the liver bound to albumin. Primarily, it's probably got some other stuff it binds to, but albumin's our primary carrier of stuff. So it's very non-water soluble. We want to be able to excrete this. This is a breakdown product, which means we need to make it water soluble because fat-soluble stuff does not get secreted real well. So we take biliverdin, which is the breakdown product of heme, we convert it to bilirubin, and then we transport it to the liver. Because the liver does most of our modification for excretion. So what the liver does is it adds a UDP glucuronate. UDP glucuronate. Still going to stumble over that one too. And liver adds a UDP bound to glucuronate. What this is called is this step is called conjugation. And please know that word. It says it right down here. Conjugation. The purpose of conjugation is to make it more water soluble. That's the definition of conjugation, not the definition of conjugation that you normally think of when you think of conjugal visits. That's not to make you more water soluble. But we're conjugating it, we're making it more water soluble, which allows us to excrete it. This happens twice. We had 2 UDP glucuronates. It goes from bilirubin to bilirubin monoglucuronate to bilirubin diglucuronide. Don't worry about the 8s and the ides. He doesn't really test over that stuff, whether it's an 8 or an ide. They do switch names, though. Now, when that happens, normally this is shipped off into the bile duct. Some of this extra stuff that I'm adding, is not stuff that Dr. Sarkar will test you on, but it is stuff you will get tested on in clinical nutrition and lab diagnosis and stuff like that. So I'm kind of giving you a primer for this stuff. So for instance, the fact that this gets shipped into the bile duct and then it's converted to urobilinogen in the intestines, not so much for Dr. Sarkar. So bilirubin diglucuronide is now water soluble. That was the point. This guy's water soluble. It's shipped into the bile duct, which means it goes into the intestines. In the intestines, bacteria turn it into urobilinogen. Most of the urobilinogen is turned into stercobilin. Stercobilin exits in the feces, and it's actually what gives feces its brown color. Stercobilin is converted to stercobilin in the large intestine or in the intestines, I don't know where. And it's what gives feces its brown color, and that's excreted in the feces. Some of the urobilinogen is reabsorbed, and it's shipped out in the urine, which is why it's called urobilinogen. So some of it is reabsorbed before it's turned into stercobilin, and it's goes back into the blood, filtered by the kidneys, excreted in the urine. This is all pending conjugation. If it doesn't go water soluble, it can't get into the bile, it can't get into the urine. It causes problems. That's it. No, you got to know that pathway. Mostly that it's turned into biliverdin and then bilirubin. Conjugation is the process of making it more water soluble. What we add is we add glucuronidates, UDP glucuronidates. Make sure you know that. And then it goes into urobilinogen and stercobilin is excreted in the feces. No enzymes, anything like that. Easy enough? Bottom of page number 103, heme is oxidized and yields carbon monoxide and biliverdin. We talked about that. That's the first step. Biliverdin is then reduced to bilirubin, transported to the liver, bound to albumin. Okay, so that might show up just in case. Albumin is a main carrier protein. If you don't know what's carrying something, just guess albumin. It reacts with glucuronate to produce a more water-soluble, and then it's conjugated with another one to produce a more water-soluble version. And again, conjugation just means makes it more water-soluble. In the intestines, it is converted to urobilinogen and then stercobilin, the pigment that gives feces its brown color. If you ever wondered why your poo is brown. Questions on the breakdown of heme? Okay, first two pathways, done, bam. And those are two big ones. Now we're going to go into creatine. Creatine is, right next to heme synthesis. So we'll go through a little bit of here. Creatine is another one of those ones that uses glycine. Who else uses glycine aside from creatine? Hemes and purine bases. Nice. Hemes and purine bases. Fantastic. Creatine is present in muscle, brain, and blood, TQ. Creatine is present in muscle, brain, and blood. Those are the only tissues that use creatine. You guys heard of creatine? Creatine phosphate, you know kind of what it does. Okay, we'll go through it a little bit more at the end. Trace amounts are normally found in the urine as you have breakdown or excretion, etc., etc. Reservoir of high-energy phosphates bonded. So you guys understand that statement? How creatine acts as a phosphate reservoir? Right here on the page number 104. Here's creatine. We're synthesizing this bad boy. What happens is in times of high energy, ATP donates a phosphate and becomes ADP, creating creatine phosphate. The enzyme that does that is creatine kinase or creatine phosphokinase, whatever you want to call it. It makes creatine phosphate. It stores the extra phosphates from that ATP on creatine phosphate, and then the ATP goes back to the mitochondria to get recharged with another phosphate and become ATP again. So now we have that ATP regenerated, and we have creatine phosphate with an extra phosphate. Then when we go use this stuff, when we're working out or when you're running from the bear or chasing hamburgers, if you're Dr. Sarkar. Sorry, Dr. Sarkar. I shouldn't have said that on podcast. You break down that ATP, you use it in cellular metabolism, in muscle contraction, in whatever it is that's going on, and now you have ADP free and floating around. Well, that wants to become ATP. It needs more energy. So what's it do? It goes back to this phosphorylated creatine, this creatine phosphate, and it reverses this reaction, as indicated here by both arrows. So creatine kinase reverses the reaction, takes creatine phosphate and makes creatine, and recharges that ATP. So it serves as it's basically another source of ATP. Why we don't just build more ATP, I don't know. But it stores an extra high energy phosphate bond. It's available for very short amounts of time. It's one of those things that weightlifters think they can get an edge by supplementing because it gives you that extra little bit of phosphorylated stuff in your muscles, so you get one last good rep out or something like that. So, reservoir of high energy phosphates bonded to it, just stores high energy phosphates when you're in times of excess, contributes them when you're in times of need. When the concentration is depleted or reduced, it regenerates it, etc., etc., etc. Same thing we just talked about. Now, TQ, creatine synthesis begins in the kidney and is completed in the liver. Begins in the kidney and is completed in the liver. Happens in 2 organ locations. Where did heme synthesis take place? Liver and erythroid cells. In what subcellular location? Mitochondria and cytosol. Good job. So, creatine synthesis takes place in both the liver and the kidney. 3 amino acids participate in it. We're going to talk about them. So up here is creatine synthesis. We'll go through this real quick. Start with glycine, right here. Glycine's important for both, TQ. Arginine. Glycine and arginine combine and they form guanidinoacetate. Now in the process, arginine is turned into ornithine. Ornithine is another amino acid, just not one of the 21 essential amino acids. So glycine and arginine combine and spit out ornithine and guanidinoacetate. Guanidinoacetate is that intermediate of creatine synthesis. That guy comes up often on tests because it's such a weird name and nobody remembers where guanidinoacetate was from. So make sure you know that guanidinoacetate is involved in creatine synthesis. Guanidinoacetate is involved in creatine synthesis. It's the precursor to creatine. That happens in the kidney. Guanidinoacetate is then converted to creatine by SAM going to SAW. What type of reaction is SAM going to SAW? I'll give you a hint. Chapter 8 coming back to haunt you. What does SAM stand for? S-adenosylmethionine. What does SAW stand for? S-adenosylhomocysteine. What's the difference between methionine and homocysteine? What does this guy do? What was the theme of Chapter 8? Oh, you guys are killing me. 1 carbon transfers. So this is a methylation reaction. SAM is a 1 carbon transfer. Remember we had tetrahydrofolate was the primary, B12 was the secondary, the last line of defense was SAM. And remember, back at the end of Chapter 8, we had this lovely little chart where I said guanidinoacetate goes to creatine by SAM. You had to know that. And I told you we were going to study it in Chapter 9. Guess what? We're also going to look over norepi and epi and nucleotides and methylated nucleotides. And acetylserotonin and melatonin. But there's a methylation reaction. So, SAM goes to SAW, methylates guanidinoacetate to creatine. I would know that it's a 1 carbon transfer. So if he asks you, what product do you get when you perform a 1 carbon transfer with guanidinoacetate? What product do you get when you perform a 1 carbon transfer with guanidinoacetate? You get creatine. Good. That's why it's a weird one, because nobody really connects those two. This happens in the liver. Guanidinoacetate becoming creatine happens in the liver. So the kidneys and the liver make this stuff, and where does it end up? Blood, brain, and muscle. Blood, brain, and muscle. What 3 amino acids are involved in this pathway? Glycine, arginine, and the one that always gets people is methionine. Because methionine is hidden in the SAM. So glycine, arginine, and methionine. Another good rule of threes. Which of these amino acids is not involved in creatine synthesis? Glycine, arginine, methionine, cysteine. Cysteine. Good. It's kind of involved, but the lame one. Or whatever. You get the idea. 3 amino acids involved. Don't forget about the methionine, sad little guy over there. released into brain, blood, and muscle. Here's your creatine to creatine phosphokinase reaction, creatine to creatine phosphate reaction. Creatine phosphokinase catalyzes it, aka creatine kinase, don't care what you call it. Also CPK or CK. Reversible ADP to ATP, it serves as a high energy phosphate storage. In times of excess, it loads up on phosphates. In times of loss, it donates those phosphates to allow the ATP stores to regenerate. Now, key little note down here. Creatine is secreted in trace amounts in the blood and the urine. They use creatine as an indicator of stuff like myocardial infarctions and muscle damage. Because if it's in excess in the blood or the urine, what does that mean? It's only in the brain, the blood, and the muscle. And it's primarily in the brain and the muscle. Primarily in the muscle. The only reason it's in the blood is to get to those guys. So it's in trace amounts in the blood. It's in trace amounts in the urine. It's in heavy concentration in skeletal muscle. Skeletal especially. So what does it say if you have lots of creatine in your urine? You have muscle damage. So it's a great indicator of myocardial infarction. It's a great indicator of rhabdomyolysis or muscular dystrophy or stuff that destroys muscles. It also can elevate if you eat a lot of red meat, because red meat's muscle, right? It also can elevate if you have a heavy workout that you're really sore from, because you've actually done minute muscle damage that's releasing creatine kinase. Or creatine. So be aware that if he asks you, what is it indicative of if creatine is elevated in the urine or the muscle, or urine or blood, it's indicative of usually muscle damage. Increase in that amount signals muscle damage, or I forgot to mention that kidney failure. Kidney failure if it's increased. Now there is, let's talk through it and I'll tell you when I get there. So, creatine, glycine, arginine, and methionine right here at the bottom. Was arginine an essential or a non-essential amino acid? Essential amino acid, good. It was the A in Private Tim Hall. Here's your methionine, I wrote it in over here. So, glycine combines with arginine, forming guanidinoacetate and ornithine, which travels to the liver, where it's methylated. Please note it's methylated by acetylmethionine for creatine, is released to the bloodstream to enter the cells. Here's that whole reaction again. I'm not going to explain it for a third time. Reaction is reversible, where it can regenerate ATP from phosphorylated creatine in times of need. Creatinine is the anhydride form. It's largely formed in muscle by an irreversible non-enzymatic degradation of the phosphate. In other words, when we break down creatine, occasionally creatine turns into creatinine. Creatinine is no longer useful. It's the breakdown product of creatine. Creatinine is excreted in trace amounts in the blood and in the urine, and that can also indicate increased damage. So free creatine is definitely bad. Free creatinine in large amounts is definitely bad. So just a breakdown product, whether it's creatine or creatinine. Depends on the amount excreted per day depends on the body's muscle mass and kidney function. Don't worry about the number. In case of kidney failure or muscle damage, it's increased. Cool? What amino acids are important for creatine synthesis? Glycine, arginine, methionine.
This is the security department. Please be advised that the creek lot and bridge gates will close at 6 p.m. Please move your vehicle from the creek lot to the main campus at this time.
For those of you watching the podcast, please move your vehicle. It's important. What amino acid is involved in heme synthesis? Glycine. How many chiral rings make up a heme? 4. What is conjugation? Make it water soluble. What liver conjugates it? What tissue conjugates? And what is the product that gives feces its brown color? Stercobilin. Nice. Which is the breakdown product of? Primarily. What are we breaking down in that whole pathway? Hemes. Good. Why would you break down hemes? Yep. What breaks hemes? What's that process called? What is the breaking of a heme called? You'll laugh when I tell you. You know it too. The breaking of a heme. Hemolysis. Remember your hemolytic streps, your hemolytic staph, stuff like that? That's breaking down heme groups. It's actually breaking down red blood cells, which therefore breaks down the heme groups inside of them. but yes, hemolysis. Okay, now we're getting into kind of the big boy in this chapter, purines and pyrimidines. These guys are on the first page. Well, I don't remember what order they are. I think it's page A, but I don't really know. We're going to start in the book, though, on page number 105, just below creatine stuff, creatine. So, dietary uptake of what are purines and pyrimidines, first of all? Nucleic acids, nice. Purines and pyrimidines are your nucleic acids, ATGC or AUGC. So, specifically, you guys need to know these. Which ones are the purines? Adenine and guanine. Adenine and guanine. A and G are your purines. We're going to talk about that and we're going to look at that, so it'll make a little more sense here in a minute. I know this is a Texas thing, but I'm from California and where I live, it's different. I always remembered Aggies are pure, like people that grow crops. Agricultural people are pure people, so it's A and G. Out here that has some sort of a weird sports meaning, so you can't say it. But however you need to remember it, A and G are your purines. I've also learned to say this differently. C, U, as well as T, are your pyrimidines. I used to say C-U-N, T, and that apparently doesn't sound like an end when I say it. So C, U, as well as T are your pyrimidines. You're never going to forget, I hope. It's probably worse the first time I said it like 10 times and people are like, do you know what you're spelling, dude? So, purines A and G, pyrimidines C, U, and T. Dietary uptake is low. They are synthesized de novo or salvaged from existing bases. In other words, if you're not eating enough of them, or if you don't, whether or not you eat enough of them, or you don't eat them at all, I don't know what that's trying to say. It sounds like he's saying they're low, but I think it's more indicative of if you're not eating them or not. We are synthesizing them de novo or salvaging. Synthesis occurs in the liver, TQ, TQ, TQ. Synthesis occurs in the liver. Brain is another area. Synthesis occurs in the liver and brain. That's what that sentence should read. Synthesis occurs in the liver and brain, primarily in the liver like every other thing, but the brain also does it. Why would the brain do it? Because the brain's important, right? Got to be able to make nucleic acids just in case. So, synthesis occurs in the liver and brain. I'll put that there for next try so I don't sound like an idiot. Liver and brain. The bases are then transported into other cells for nucleic acid biosynthesis. All right, give me your sheet. You can TQ these right now if you want on this sheet, actually. Precursors, glycine, glutamine, and aspartate. Glycine, glutamine, and aspartate. 3 amino acids, rule of threes. Glycine, glutamine, aspartate. So those other guys are nice, and we'll talk about them a little bit, but they're not specific TQs. And you're going to kind of know some of them. So purine synthesis is on the left side of the sheet. It says right at the top of the sheet, purines and pyrimidines are synthesized de novo or salvaged in the brain and liver. And right under here it says two very important things, and I need you to highlight these or write these or whatever. In purine synthesis, the base is built on the sugar. What sugar do we use for nucleic acids to make DNA or RNA? Ribose or deoxyribose. But ribose is the sugar we use, which is how many carbon sugar? 5 carbon sugar. So we use ribose. What this means is that the first thing we do in purine synthesis is we take a ribose and we build the base around it. And when I say base, we're talking about the nitrogenous base. And remember that a nucleoside with an S, a nucleoside with an S, is a base and a sugar. The difference between a nucleoside and a nucleotide with a T is it's now a base and a sugar and a phosphate. So, nucleosides, base and sugar, nucleotides, base plus sugar plus phosphate. So, in purine synthesis, the base is built around the sugar, which means the first thing we do is we take a sugar and we start building the base around it. In pyrimidine synthesis, we'll talk about this a little later, the base is built first and then it's attached to the sugar. You guys get the key difference. In one of them, we take the sugar, we use it as the foundation. In the other one, we build the house and then we slap it on the foundation. Make sure you know that. That'll come up. One of those two. True or false? In pyrimidine biosynthesis, the base is built on the sugar. You got to know that's false. Because in pyrimidine synthesis, the base is built first and attached to the sugar. In purine biosynthesis, the base is built on the sugar. We start with our ribose 5-phosphate. It's got a convenient little note on here. It comes from the HMP shunt. Pentose phosphate pathway. That's important. He may ask you about that. The source of ribose 5-phosphate is the HMP shunt or the pentose phosphate pathway. Probably won't say pentose phosphate pathway. That's a little obvious. Ribose 5-phosphate is turned into 5-phosphoribosol 1-pyrophosphate. It's a terrible name. Very easy though. Ribose 5-phosphate is a ribose sugar and 1-phosphate. We are taking ATP and turning it into AMP, which means that we are gaining 2 phosphates. So down here, we have 5-phosphoribosol 1-pyrophosphate. Ribose and 1-phosphate, ribose and 1-phosphate. And then we're taking 2 from this ATP and attaching them right here. Remember, a pyrophosphate is just 2 phosphates attached together. That guy is 5-phosphoribosol pyrophosphate, aka PRPP. That's most commonly what you're going to see it as is PRPP, 5-phosphoribosol pyrophosphate. All you have to do is count the phosphates. 3 phosphates. Bam. You know where you are. Now, the guy that does that is PRPP synthetase. What does synthetase say it's doing? Why is it a synthetase and not a synthase? Because if it's a synthetase, it's using energy. It's using ATP. If it was just a synthase, like delta ala synthase, that one follow my rule? Yep. Delta ala synthase has no ATP involved, so it's just a synthase. If it is a synthetase, you know there must be ATP involved. Another little biochem thing that helps you out a little bit. So PRPP synthetase, no big deal. Named for its substrates because it's named for its products because it's technically its substrates are ATP and ribose 5-phosphate. So we're making PRPP. PRPP. Now there's a lot of steps in between all this stuff that you don't really need to know. What's important is that the rate-limiting enzyme, which happens first, adds a glutamine, which is one of those TQ amino acids you need to know. It takes PRPP, which is your sugar, because we're building the base on our sugar in purine biosynthesis, and it adds a glutamine to it. The name is terrible, and it's called one of 2 things. It's glutamine phosphoribosol aminotransferase, glutamine phosphoribosol aminotransferase, or PRPP glutamyl aminotransferase. If you see glutamine or glutamyl and PRPP, it's your rate-limiting enzyme of purine biosynthesis. It's going to have both of those bad boys in it, whichever one he chooses to use on the test. It's going to say PRPP and it's going to say glutamyl or glutamine in some way. That's your rate-limiting enzyme for this. Now there's a bunch of steps leading to where we're going next. We don't care about most of them. We're adding glutamine, we're adding glycine, and we're adding aspartate. Those are our 3 amino acids you got to know for this bad boy. Be aware that there is also N10 formyl FH4. What is that? N10 formyl FH4. What's FH4? Tetrahydrofolate. What's tetrahydrofolate do? Chapter 8. What's chapter 8? 1 carbon transfers. You guys are terrible. Those of you at home are even more terrible because none of you answered. I mean, at least these 3 tried. 3, mind you. I went from 60 to 3. You're all fired. So, 3 amino acids you got to know. Glutamine, glycine, and aspartate are the 3 amino acids that we add to make a purine base. There is also some methylation going on because formyl FH4 is a methyl donor. There's some carboxylation, there's some methylation. So there's some other stuff going on, but they're not your major points for this section. They do other stuff in the process, but big 3 is we add these 3 amino acids. And you'll notice glutamine's added twice. Doesn't matter. Not going to ask you that. But glutamine, glycine, and aspartate are your big 3 right here. Now you do need to know this. When all of this is combined, when we've built our base on top of our sugar, we form inosine monophosphate, IMP, which is the first nucleotide in purine synthesis, TQ, TQ, TQ. First nucleotide in purine synthesis is IMP. Do we use IMP for DNA or RNA? No, but it's the first nucleotide we make. Why is it a nucleotide? Because it has a nitrogenous base, it has a sugar, and it has a phosphate. If it was just IM, it would just be a nucleoside. But since it's got that phosphate attached, it is a nucleotide. First nucleotide in purine synthesis is IMP. Now here's where we get fun. What are our two purine bases? A and G. So we got to make A and G. Guess what? We can make A and we can make G. So to make A, we take inosine monophosphate and add an A. We add an aspartate. We're already using aspartate. It's one of those big three you got to know for purine biosynthesis: aspartate, glutamine, and glycine. But we just take inosine monophosphate, our first nucleotide in purine synthesis, and we add an A to it. If we want to make the G, we take IMP and we add a G to it. Now these guys require an energy source. So to make the A, we take aspartate, but we use GTP. Why would we use GTP over there and ATP over there? The purpose of these pathways is to synthesize these bases. You would synthesize these bases if you were deficient in them. So, if we were deficient, oh wow, I'm way off the chart, sorry. If we were deficient in A, which we've made with aspartate, we wouldn't want to rely on ATP as an energy source because we're deficient in it. So we're going to rely on GTP as an energy source, just as a backup. If we want to make a GMP, we're going to add some glutamine, G and G, but we don't want to rely on GTP as an energy source because we're making Gs. So we're going to rely on ATP as an energy source. It's just a backup mechanism of redundancy in case, for some reason, you're completely out of it, then you couldn't make it, and that would be a vicious cycle. So, either way, we make these guys by adding one of those three main amino acids you got to know, and then we phosphorylate them. This guy is called nucleoside monophosphate kinase because he takes a nucleoside monophosphate, aka a nucleotide, and he kinases it to a D version. No big deal, name for what he does, not an important enzyme. Now, nucleoside diphosphate kinase takes the diphosphate versions and kinases them, making the triphosphate versions. Those guys can go off to RNA synthesis. Those guys can go right off to RNA synthesis. You guys following along so far? Not confused? ATP, GTP, bam, RNA synthesis, because we use A's and we use G's for RNA. But we don't use ATP and GTP for DNA synthesis. We use deoxy ATP and deoxy GTP. We use the deoxidized versions of the ribose sugar, which we have to make. There's a very important enzyme you got to know that makes these bad boys. That enzyme is ribonucleotide reductase. Ribonucleotide reductase reduces these guys. What does reducing mean? He's deoxying them. Because remember, the loss of oxygens is reduction. The addition of oxygens is oxidation. The loss of oxygens is reduction. So if we're losing oxygens, we're being reduced, which is why he's named ribonucleotide reductase. Ribonucleotide because he's a nucleotide attached to a ribose sugar. Ribonucleotide reductase. Important, important, important TQ. Ribonucleotide reductase only works on the diphosphate versions. He cannot ribonucleotide reduce AMP. You cannot ribonucleotide reduce ATP. You can only ribonucleotide reduce ADP, which is why it takes them from this chain. Ribonucleotide reductase produces deoxy ADP, deoxy GDP, because it only works on the diphosphate versions, and those guys go off to DNA synthesis. You guys cool with that? One last TQ. Ribonucleotide reductase is an iron-containing enzyme. Therefore, iron deficiency causes deficiency in nucleotide biosynthesis. Iron deficiency means you can't produce your nucleotides. Nucleotides are pretty important. Iron deficiency means you can't produce your nucleotides, and those bad boys are important. And it's all because of ribonucleotide reductase. So, what's going to be primarily affected? DNA or RNA? DNA, right? Why? Because ribonucleotide reductase makes the D versions, which are only important for DNA. We have plenty of ADPs and GTPs that we can make right up here for RNA synthesis, but we can't make the D versions, so we can't make DNA. Now, if you don't have DNA, you really don't need much RNA kind of a moot point. But just interesting that iron deficiency will cause heme deficiency, and it will also cause DNA or something like that deficiency because you can't make it. So, last little notes. Any questions on the synthesis pathway? What three amino acids? Glutamine, glycine, and aspartate. True or false? In purine biosynthesis, the base is built first and attached to the sugar. True or false? In purine biosynthesis, the base is built first and attached to the sugar. False. The base is built on the sugar. Good job. What's the first nucleotide in purine biosynthesis? IMP. IMP is the first nucleotide in purine biosynthesis. What mineral ion is important for ribonucleotide reductase? Iron. Did I say iron? No. Okay. Random TQs. So, lastly, when nucleotides are broken down, the purine bases can react with PRPP to regenerate it. In other words, when these bad boys break down, you break it into a phosphate, you break it into a nitrogenous base, and you break it into a ribose sugar. When these guys are broken down, we can salvage that nitrogenous base, attach it to a PRPP, and have our nucleotides back. So we can take the A that's just floating around, adenosine or adenosyl that's just floating around, slap a PRPP on it, now we've got adenosyl triphosphate or adenylate triphosphate. So we can get these guys back. We can salvage them if they get broken down to regenerate the nucleotide. There are two enzymes that do this, not big TQs. I would more know the abbreviations than anything, and even that would be a stretch. Hypoxanthine guanine phosphoribosal transferase, aka HGPRT, and adenine phosphoribosal transferase, aka APRT. This guy transfers the G's, this guy transfers the A's. Again, not huge points. What you do want to know, and even this is kind of whatever, is that Leach-Nyland syndrome is defective HGPRT. You can't resalvage these. If you can't resalvage them, they sit around and form xanthine. And xanthine is converted to uric acid, which causes gout and mental retardation. So Leach-Nyland syndrome is a genetic deficiency of the salvage enzymes. If you can't salvage these, every time these bad boys are broken down, they sit around, they turn into xanthine. Xanthine is converted to uric acid and causes uric acid buildups, which is gout, and it can cause mental retardation. Whew. You guys good? Need a 5? Or you want to power through? Okay, we'll take a couple minutes. Or I'll just keep talking and you'll have to listen from, just go to the first stall, the one closest to the room. Bathroom break on vodcast. Go pee now, or pause it if you want. apparently try 6 is easy street. All right, so we took care of purine biosynthesis, pyrimidine biosynthesis. Oh, I guess let's flip back through the book real quick. So we talked about this. Glycine and glutamine and aspartate are your big 3 amino acids you got to know. PRPP, glutamyl aminotransferase, a.k.a. glutamine phosphoribosal aminotransferase, however you want to call it. That's the rate-limiting enzyme. Negative feedback is your primary thing, but he doesn't really talk about it. All this stuff, don't really worry about it. It's all those things. We talked about some methyl donations. We talked about some glycine. We talked about some carboxylation. All that stuff's important, but not really specific TQs. You do want to know the first nucleotide is inosine monophosphate, TQ, first nucleotide in the purine synthesis pathways, IMP. Rapidly transformed and kinased and et cetera, and we're done. So, you guys good with that? All right. This is that pathway. Glutamine phosphoribosal aminotransferase rate-limiting enzyme, all the stuff all drawn out. Down here at the bottom, ribonucleotide reductase is an iron-containing enzyme. It says that later in the chapter. Conversion of ribose sugar to deoxyribose. It works on the diphosphate form only. We talked about that. Purine bases can be salvaged by re-reacting with PRPP. In other words, when we break them down, we can slap another sugar on the base, and we have ourselves a nucleotide again if we need it. They're salvaged by these enzymes. This is the clinical correlate Lesch-Nyhan syndrome, is defective HGPRT. Buildup of xanthine, which is the breakdown product, turned into uric acid, causes gout and mental retardation. Bam. Okay. Pyrimidine synthesis. Aspartate and glutamine. What are we missing? Glycine. Because glycine is involved in what? Give me some names. Purines, hemes, and creatines. Glycine is involved in purines, hemes, and creatines. In purines, you also have glutamine and aspartate, but in pyrimidines, you just have glutamine and aspartate, no glycine. So same thing as pyrimidine, you just ditch the glycine, and you know there's no glycine because I made you memorize that glycine is in hemes, creatines, and purines only. So, you're set. There's your 2 donors. Those are where a lot of your TQs come from this chapter, is what amino acids are involved in what pathways. We can talk about this real quick. It's on the chart. Pyrimidine nucleotides, the base is synthesized first, then it's attached to the sugar. That's your big TQ difference. The base is synthesized first, and then it's attached to the sugar. Really shouldn't have highlighted in these colors. It doesn't come up real well on that screen. What if we do the thing? Bam. Now it comes up. All right. I love doing that. First time I figured it out, it scared the crap out of me. I was like, oh my God, I broke it. So we're in the top right of that sheet. It says pyrimidine biosynthesis right up there. Here's that little note that the base is built first and then attached to the sugar. Make sure you know that. We're going to take our glutamine. Our glutamine is our starting point. And we turn it into carbamoyl phosphate. Oh, where else have you seen carbamoyl phosphate? Don't look at your sheet. Don't you read my scribbles back there?
I can't read your scribbles.
Where else is carbamoyl phosphate? Urea cycle. Remember how I told you we had that rate-limiting enzyme of urea cycle, which was called mitochondrial carbamoyl phosphate synthetase 1? Mitochondrial carbamoyl phosphate synthetase 1. Well, now we have cytosolic carbamoyl phosphate synthetase 2. Got to know it. Carbamoyl phosphate is also used in the urea cycle. I made a note of it because occasionally that shows up on a TQ. That where is the intermediate? What common intermediate is shared in pyrimidine synthesis and urea cycle, or some weird worded question about carbamoyl phosphate. They also have a very similar enzyme. It's probably an isoenzyme, which means it's pretty much the same enzyme. It's just in a different tissue, has different rate-limiting allosteric regulation, etc. Cytosolic carbamoyl phosphate synthetase 2. It's the rate-limiting enzyme of pyrimidine biosynthesis. Almost the same enzyme that you have in urea. Urea is mitochondrial carbamoyl phosphate synthetase 1. Please do not be fooled if he puts on the test CPS1 and CPS2. CPS2 is the one we're using here. You know that because this is a later chapter in the book and we're going sequentially. CPS1 is used in the urea cycle, chapter 7. CPS2 is used in chapter 9. So it's later. Make sure you know it's CPS2 just in case he throws CPS1 in there as a distractor. It is the rate-limiting enzyme. We're going to skip the activation and inactivation until we get to the end of the end of this section. So we make carbamoyl phosphate. from glutamine and some ATPs and CO2s. Where do we get our CO2s from? Bicarb. Donations of all carboxylations come from bicarb. If you see carboxylation, it's coming from bicarb because we don't have free CO2 floating around. So, glutamine, first nucleotide. What other nucleotide or what other amino acid are we looking for? Glutamine and aspartate. You guys ever watch Super Troopers? No. Oh, God, it's a great movie. Sorry. Carbonyl phosphate is combined with aspartate. It forms orotate. Orotate is kind of that nitrogenous base that we're looking for. We're going to attach it with the sugar because the base is built first and then attached to the sugar. So here's the base, here's the sugar. We combine them together and we get UMP. Involves decarboxylation. What vitamin decarboxylates? PLP, pyridoxyphosphate, aka vitamin B6. UMP is the first nucleotide in pyrimidine biosynthesis. Important. What's the first nucleotide in purine biosynthesis? IMP. The first nucleotide in pyrimidine biosynthesis is UMP. Now this is really easy to remember. What are your pyrimidines? What? CUT are your pyrimidines. Which means that UMP dang well better be in pyrimidine biosynthesis because it's one of the pyrimidines. So if you're confused about the I's and the U's, the U goes with the U. The U goes in pyrimidine biosynthesis. So long as you know that CUT are your pyrimidines, you know that the U is the first nucleotide in pyrimidine biosynthesis. The I doesn't really involve anything else, but it is in purine biosynthesis. It doesn't match up with anything. So first nucleotide in pyrimidine biosynthesis is UMP. UMP is phosphorylated until it gets to UTP, and guess what? It goes off to RNA synthesis. It is then converted to CTP and goes off to RNA synthesis because we're looking for a C, a U, and now we got to find a T. So we got to get all the way to T, which is the funky part. It goes through all these different steps. It goes down to, so now we have our RNAs, right? We have A and G, and we have U and C, because RNA is AUGC. Bam, we're done with RNA. RNA is synthesized. We need to make T's still. We need to make the pyrimidine T. What is the pyrimidine T used for, RNA or DNA? DNA, right? Because RNA is AUGC, DNA is ATGC. So all we're missing now, that's a C, is the T, and that's what we got to make. So we take our C, we drop it down to a CDP, and then we ribonucleotide reductase it. Why do I have to drop it down? Why do I have to dephosphorylate it once? Because ribonucleotide reductase only works on the diphosphate versions. Bam, nice job. So it takes it from CDP to DCDP because ribonucleotide reductase, which is a what requiring enzyme? What? Iron requiring enzyme, only works on the D versions, the diphosphate versions. It's then converted to DUMP, and then DUMP goes to DTMP. You saw this before. DUMP goes to DTMP. It's a THF or an FH4 or a tetrahydrofolate donor, which means it's being methylated by tetrahydrofolate. The enzyme is thymidylate synthase. Do you remember this reaction from Chapter 8? Dump goes to DTMP by thymidylate synthase. The donation comes from a methyl tetrahydrofolate, so one methyl donor. It's blocked by 5-fluorouracil. If you have any questions, go back to page number, way the hell back here. Sorry, no cussing on vodcast. Page number 93, that's this reaction right here. DUMP to DTMP. Thymidylate synthase blocked by 5-fluorouracil uses tetrahydrofolate as a methyl donor. Okay, tetrahydrofolate is a methyl donor. That was what we talked about at the end of Chapter 8 when it talks, well, never mind, sorry, I'm lying. Ignore me. So, DUMP to DTMP, tetrahydrofolate, which is why folate is important for DNA synthesis. DTMP gets phosphorylated, now it goes to DNA synthesis. And I forgot to mention up here, here's your DCDP that goes to DNA. So now we've synthesized all of our DNAs and RNAs. Questions on any of that? No? You get comfortable with the amino acids that are in them all? Comfortable with the rate-limiting enzymes? Ribonucleotide reductase, iron-requiring enzyme, works on the diphosphate version only. First nucleotide in pyrimidine biosynthesis is UMP. First in purine biosynthesis is IMP. What do we conjugate bilirubin with? UDP gluconorate. Conjugate it with UDP gluconorate. And what's the purpose of conjugation? Make it more water soluble. We talked about conjugation before too. We talked about it. Do you guys remember what the heck was that? Is that cholesterol biosynthesis? When we talked about taurocolic acid and we took cholic acid and quinocholic acid and we conjugated them with amino acids. We made them more water soluble. So it's the bile salts and that process. Conjugation, make it more water soluble. All right. Pyrimidine nucleotides, bases synthesized first, then added to the sugar. Make sure you know that. Rate limiting enzyme. Oh, that's what I got to talk about. Back on the sheet, in the top. Rate limiting enzyme is inhibited by UTP. Why is it inhibited by UTP? Because it's a product. Fantastic. Rate limiting enzyme is inhibited by products and activated by substrates. PRPP isn't really a substrate of the rate limiting enzyme, but it's a substrate of the pathway, right? If we have lots of PRPP, it's saying, hey, we got all this sugar hanging out, let's make some nucleic acids. So it's a substrate of the pathway. It's not a direct substrate of the enzyme itself. But you don't really need to memorize them because you know they're right there. All right. Rate limiting enzyme in the pyrimidine biosynthesis is cytosolic carbamoyl phosphate synthetase 2, which is a very similar enzyme to mitochondrial carbamoyl phosphate synthetase 1, which is in the urea cycle. That should probably be synthetase. It's inhibited by UTP and activated by PRPP. It's inhibited by products, activated by substrates. We add an aspartate because it uses aspartate and glutamine, and it started with glutamine. Orotate reacts with PRPP to produce the first nucleotide, uridine monophosphate, or UMP, first nucleotide in pyrimidine biosynthesis. Converted to yada, 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 rate-limiting enzyme. We talked about all of this. First nucleic acid, or nucleotide. It's ribonucleotide reductase, and then it comes way over here. Gets thymidylate synthesized by THF, tetrahydrofolate, which is a methyl donor, which is blocked by 5-fluorouracil. And it's converted to DTTP for DNA. Cool? Ribonucleotide reductase is an iron... Oh, here we go. Ribonucleotide reductase is an iron-requiring enzyme, iron deficiency, that can thus cause decreased nucleotide biosynthesis, affecting primarily DNA or RNA. DNA, because you only need to reduce or deoxy the DNA versions. The RNA is preserved because you don't need to do that at all. All right, you guys are troopers. We're almost out of here, kind of. Kind of. It's a long chapter. All right. We're on page number 110, and we're switching gears. We talked about tyrosine. We talked about glutamine. We talked about aspartate. We're done with those guys. I'm sorry. We talked about glycine. We talked about glutamine. We talked about aspartate. We're done with those bad boys. We're switching gears to tyrosine. Page number 110 is an overview of everything that tyrosine does. Tyrosine makes dopamine, norepinephrine, and epinephrine. These are your biogenic amines, a.k.a. your catecholamines. Please know those words. Biogenic amines and catecholamines. Catecholamines is more commonly used. But if you ask you what amino acid is the precursor of your catecholamines, you got to know your catecholamines are dopamine, epi, and norepi, and those come from tyrosine. Tyrosine also produces melanin, which we're going to talk about. Melanin is what? Skin pigment, good. And it produces your thyroid hormones, and he loves thyroid hormones. He does research on them. So please know your thyroid hormones. We're going to talk about those. Those are fun. So, precursor of your thyroid hormones, your skin pigment, and your catecholamines. First up is catecholamines. We are on the third page, the little skinny one. The very back, it should say something about it at the top, but you can write it in if you want. So we're dealing with this. This is catecholamine synthesis. I don't know why this thing has to be so low to get on the screen. No. Ah, nice. I'm smart. Say it. So, we start with phenylalanine, and we go to tyrosine. But I thought you said tyrosine, not phenylalanine, right? Tyrosine is the precursor to these bad boys. But tyrosine is not a nutritionally essential amino acid. Phenylalanine is. Why is phenylalanine an nutritionally essential amino acid and tyrosine not? Because we can't synthesize phenylalanine, but how do we synthesize tyrosine from phenylalanine? How do we make this? What's the difference between phenylalanine and tyrosine? What's the difference between fat allen from try one and fat alcoholic allen? You got an alcohol, right? They both have benzene rings. They're both those fat old boys with benzene rings, which you can see right here. Oh, it doesn't show you phenylalanine. Here's tyrosine right here. The only difference between tyrosine and phenylalanine is that phenylalanine doesn't have that terminal alcoholic group on there. That's just fat allen. Fat allen doesn't have the alcoholic group. He's not fat alcoholic allen yet. We already gave up alcohol. So, to get our phenylalanine to tyrosine, all we have to do is hydroxylate it. We have to add a hydroxy group to it. So we use a very easy enzyme. We use phenylalanine hydroxylase, because the only difference between phenylalanine and tyrosine is a hydroxyl, or is a hydroxy side group. So phenylalanine hydroxylase it. Does anyone remember what BH4 stands for? No? Tetrahydrobiopterin. Tetrahydrobiopterin. That was part of that weird chapter where we talked about, I don't even remember, beginning of chapter 8 maybe? Maybe. We said THF, tetrahydrofolate, is a methylator or a one-carbon donor. BH4, or BHF, or whatever you want to call it, BH4, is tetrahydrobiopterin. Its sole purpose as a coenzyme is ring hydroxylation. Tetrahydrobiopterin is responsible for ring hydroxylation. So anytime we hydroxylate a ring structure, anytime we add an alcoholic group to a ring structure, we use BH4. In other words, phenylalanine hydroxylase is hydroxylating a ring, it uses BH4. Go from phenylalanine to tyrosine. To go from tyrosine to dopamine, from tyrosine to L-dopa, is only L, not like L as in Spanish, dopa. It is L as in levorotatory dopa. Tyrosine to dopa is tyrosine hydroxylase. We're hydroxylating the ring structure. What do we need? BH4, tetrahydrobiopterin, which is ring hydroxylation. So guess what? On this little sheet right here, we're going from tyrosine to tyrosine with an extra ring attached, or an extra OH group attached to it. Ring hydroxylation requires BH4, tyrosine hydroxylase. First two enzymes, really easy. Phenylalanine hydroxylase, tyrosine hydroxylase. Now, this chart gets a little splitty, so we're going to talk about this pathway first. So, phenylalanine goes to tyrosine. Tyrosine is transported through the bodies, and it's picked up by melanocytes. Melanocytes are present in your skin. It's picked up by melanocytes where it tyrosine hydroxylases, converts to dopa, and then converts to melanin. Melanin is one of your products of tyrosine. That happens in melanocytes. Now, not in melanocytes, tyrosine is shipped out into your blood, and it's picked up by neurons. Neurons, tyrosine hydroxylase, and then dopa. Now, the reason I didn't just branch melanin off of here is because technically dopa isn't shipped around the body. It's tyrosine that's shipped around the body, and then these tissues just happen to have the same enzyme. Does that make sense? So, tyrosine's shipped around the body wherever it needed, or phenylalanine is, and it's phenylalanine hydroxylase. Melanocytes take it to dopa and then to melanin. From tyrosine to dopa is tyrosine hydroxylase, ring hydroxylation, coenzyme BH4, tetrahydrobiopterin. From tyrosine to dopa is tyrosine hydroxylase, BH4, tetrahydrobiopterin. Now we continue on. This guy goes to melanin, but down here we take the dopa and we dopa decarboxylase. We DD it, dopa decarboxylase. What enzyme is responsible for what coenzyme is responsible for decarboxylation? I'm falling to the ground. PLP. PLP is responsible for decarboxylation. I can't blame you guys yet. You haven't had Chapter 11. Chapter 11 is where you have to know that stuff, but you got to know it on the next test, and I'm trying to prime you on one of the 12 you got to learn. So, dopa decarboxylase is a decarboxylase. Since it's a decarboxylase, we are losing a carboxyl group, And the enzyme is PLP, pyridoxal phosphate, vitamin B6, which does all decarboxylation and all transamination. Should I ask you the transaminations? No. Oh, God, make sure you know those. So, dopa decarboxylase, PLPP, vitamin B6 decarboxylation, goes from dopa, or L-dopa if you prefer, to dopamine. Now, this is really cool. I'm going to lose my sheet. So, you guys aren't going to think it's cool, but I think it's cool. Phenylalanine to tyrosine, ring hydroxylation. Tyrosine to L-dopa, ring hydroxylation. What's the difference between an amino acid and an amine? An amino acid and an amine. We lose the acid, right? So how do you get from an amino acid, an L-dopa, to an amine is by ditching the CO2. And what do we lose? Is we lose this guy right out here. It's no longer an amino acid, because here's the alpha carbon of the amino acid. It's got a carboxyl group, it's got an amine group, and it's got a side chain. It's an amino acid. If we ditch that amino acid by decarboxylating it, it's no longer an amino acid, it's just an amine. It's just an alpha carbon with an NH3 attached to it. You guys see that? So to go from dopa to dopamine is just decarboxylation, because that's how you go from amino acid to amine. So the enzyme is dopa decarboxylase. Dopa decarboxylase decarboxylates it, becomes dopamine. And then to get from dopamine to norepinephrine, we take dopamine beta hydroxylase. That's an gnarly one. Dopamine beta hydroxylase. Hydroxylates the beta position. You can see that on your chart in your book. Dopamine beta hydroxylase. Hydroxylates the beta position right here, alpha beta, making norepinephrine. Now please note, he has this in the book as well. Copper and vitamin C are both required for this process. I wrote it on my chart. Copper and vitamin C are both responsible for this process. Dopamine beta hydroxylase. Norepinephrine. Bam, that's done. That's where you end up in neurons. Neurons of the sympathetic nervous system and other places, locus coeruleus and stuff, can make norepinephrine and secrete norepinephrine as a neurotransmitter. There's only one place in the body that makes epinephrine. That is the adrenal medulla. So, just like we did for the melanocytes, I didn't want to put another chart on here. In the melanocytes, you take tyrosine, you turn it into melanin. In the neurons, you take tyrosine, you turn it into norepinephrine. Now, if we were to erase the neurons from that, in the adrenal medulla, we take tyrosine, we turn it into epinephrine. Norepinephrine isn't shipped out to be turned into epinephrine somewhere else. They're powerful neurotransmitters. They each do their own shipping out. Cool? So each tissue takes tyrosine, or phenylalanine and tyrosine, and makes its own pathway of stuff and then secretes it as needed. But tyrosine or phenylalanine is what's safely shipped around the body and transported into other stuff. To get from norepinephrine to epinephrine, you had to learn this one last chapter, SAM to SAW. What type of reaction is that? Oh, thank you. One carbon transfer. I don't have to commit harikari. Is that Japanese suicide? Or samurai suicide? I don't know if it's harikari. Norepinephrine to epinephrine is SAM to SAW. It's at the end of chapter 8. It's one of those little things you had to memorize. This is a fun one. Phenylethanolamine N-methyltransferase. Phenylethanolamine N-methyltransferase. If you look at your structure, it makes a lot of sense. So we beta hydroxylase, we got this. Now we're going to take a phenyl ethanol amine. A phenyl ethanol amine. And we're going to N-methyltransferase it, which means that we're going to methylate it at the N position. That's at the amine position. Remember, N-linked glycosylation and O-linked glycosylation, or not glycosylation, but methylation in this process. So phenyl ethanol amine N-methyltransferase. Saying what it is, a weird enzyme. You're probably going to be able to pick it out on test if you ask me about it, because it's such a weird enzyme. We make epinephrine. I would know that it requires S-adenosylmethionine. It's a one-carbon transfer reaction. We talked about that in Chapter 8. He loves to hit stuff like that. And according to Ian, although it didn't show up on our boards, it's a big one on boards. All right. Questions about catecholamine synthesis? Cool. What amino acid was that? Not glycine. Fantastic. Not glycine, not aspartate, not glutamine. It was tyrosine. which comes from what nutritionally essential amino acid? Phenylalanine. Fantastic. You said it. I just didn't process fast enough. Now, I've got some good news for you. This is your synthesis pathway for thyroid hormones, and now that I'm looking at it, it's really not quite correct. So we're going to change some stuff. It's mostly correct. So we're going to do this. I'm going to put a big arrow over there, and we're going to say add a DIT. And you'll know what I mean here in a second. And we're going to change this arrow to go this direction. We'll talk about all this. Or you can go this way, and you can add a MIT. That doesn't make much sense, but we're going to have some fun with it. Good news is that's really all you need to know. And primarily, you don't really even need to know that so much as you need to know that thyroid hormones come from tyrosine, because we don't talk about any enzymes, we don't talk about anything else. So, once you've made those changes, go ahead and look up for me, and we're going to have some fun. So, we take tyrosine. All you need to know for this is we take tyrosine and we iodinate it, which is why these bad boys are called monoiodotyrosine. So what you want to know is that tyrosine is important for thyroid hormone synthesis, and iodine is important for thyroid hormone synthesis. We take tyrosine and we iodinate it. We get monoiodotyrosine. That's a MIT, M-I-T, monoiodotyrosine. We take that monoiodotyrosine and we iodinate it again. We get diiodotyrosine. That's a DIT. If we take a MIT and a DIT, how many iodines do we have? 3. What does that make? It makes a TIT. Monoiodotyrosine but plus diiodotyrosine makes a TIT. Tyrosine, which is what this bad boy is saying up here. We take tyrosine, we iodinate it. We get monoiodotyrosine. We iodinate it again. We get diiodotyrosine. And then what happens is we add a MIT to our DIT, and we get a TIT. We get triiodo. It's no longer tyrosine, and I'm going to show you why here in a minute. It's triiodothyronine, a.k.a. T3. 3 for how many iodines it has attached to it. Or contrarily, we can take a DIT, and we can add another DIT, and we get another form of TIT. We get diiodotyrosine plus diiodotyrosine makes tetraiodothyronine, T4, because it has 4 iodines attached to it. This is a.k.a. thyroxine. Now, last question. How do we get from tetraiodothyronine to triiodothyronine? We take what off? An iodine. Not a full MIT, just an iodine in this case. So now what I'm going to show you is what he has drawn over here. Here's tyrosine right here. Here's the alpha carbon, here's the amino group, here's the carboxyl group, here's the side chain of tyrosine. We take tyrosine, we add an I to it, we add an iodine to it. We get MITs, monoiodotyrosines. We add another iodine to it. This is 3', this is 5, this is the 3rd position, this is the 5th position. That's why it's called 3-monoiodotyrosine, 3,5-diiodotyrosine. There's still tyrosine, we've just iodinated it at 2 positions. Now here's the fun part. To get a TIT, to go from diiodotyrosine to triiodothyronine, we don't have any more room for iodines on here. So we attach a whole other benzene ring right here. We rip it off another tyrosine. So we take a full tyrosine plus a DIT, and we add the benzene ring with 1 iodine attached to it to get a TIT. So 3, 5, 3 prime, and you don't need to know the 3's and the 5's and primes. Triiodothyronine. It is no longer triiodotyrosine because it's no longer tyrosine. As soon as we add this to it, it's not tyrosine. It's thyron. This is T3. That's your active form. Probably don't even need to know that for Dr. Sarkar. Did he go real in depth on these boys? Okay. T3 is your active form of thyroid hormones. Or if you take diiodotyrosine and you add a diiodotyrosine to it, you get tetraiodothyronine, which is 4 iodines attached to it. And how would you get from here to here? You'd just take this iodine off. So, primary points. Tyrosine is the precursor of your thyroid hormones. It requires iodination. Your AKAs, did you have them written here? Your AKA for T4, or tetraiodothyronine, is thyroxine. I would know that. And your AKA for T3 is triiodothyronine. That's it. You're going to go into a lot more detail on the thyroid hormones in physio, I think. But this is all Dr. Starkar wants you to know about them. Primarily, the stuff that's going to come from it is iodine requiring and tyrosine precursor. Cool? All right. We've covered everything that tyrosine does. Tyrosine does thyroid hormones. What else? Catecholamines, and one more thing. Skin pigment, melanin. Tyrosine does catecholamines, skin pigment, and thyroid hormones. Now, I think this is the last big pathway, and then we have like 3 small pathways, and we're almost done. We're going to talk about tryptophan. Now, people always get confused between tyrosine and tryptophan. Please keep them straight. Tyrosine and tryptophan. Tyrosine makes your catecholamines. Tryptophan makes your onins, serotonin and melatonin. What is melatonin? It's a neurotransmitter involved in the circadian rhythms. It's not melanin. Melanin and melatonin are different. Melanin is made by tyrosine, and it's your skin pigment. Melatonin is a neurotransmitter involved in circadian rhythms, aka the sleep-wake cycle. Melatonin is a neurotransmitter involved in the sleep-wake cycle. It says it right here. Produced in the pineal gland, regulates circadian rhythm, or the day-night cycle, or the light-dark cycle, or the sleep-wake cycle, whatever you want to call it. It's all listed right here. But tryptophan makes your onins. Tryptophan makes serotonin and melatonin. It also generates the cofactors nicotinamide nucleotides. We're going to talk about that. So, on your sheet in the bottom right corner from heme biosynthesis, we are talking about tryptophan. First of all, tryptophan makes the nicotinamide portion of NAD or NADP. What that means is when you have NAD and NADP, that NAD, that nicotinamide portion, came from tryptophan. We're going to talk about that. NAD is vitamin B3. So tryptophan is involved in the synthesis of NADs and NADPs. It makes the nicotinamide portion, TQ. It makes the nicotinamide portion of NAD and NADP. Those words. Tryptophan makes the nicotinamide portion of NAD and NADP. Tryptophan is acted on by BH4. What are we doing to tryptophan? Yes, we're ring hydroxylating it because tryptophan is an aromatic amino acid. We're ring hydroxylating tryptophan. We're getting 5-hydroxytryptophan. Tryptophan to 5-hydroxytryptophan involves ring hydroxylation, which means it must be a BH4 coenzyme. And then it is PLP'd. PLP is vitamin B6, and it does what? Decarboxylation and transamination. 5-hydroxytryptophan is decarboxylated. What happens when we take... I can't say that. Yeah, I can. Where are we? Tryptophan right here. Nicotinamide portion of NAD, NADPH, or NADP. BH4 does ring hydroxylation. We get 5-hydroxytryptophan. It's decarboxylated. What happens when we take an amino acid and we decarboxylate it? It becomes an amine. Serotonin, and this is the way you're going to want to know it, is most often now called 5-hydroxytryptamine. 5-hydroxytryptamine, or 5-HT. 5-hydroxytryptamine is usually how boards asks it. Its AKA is serotonin. And it's just the decarboxylated version of 5-hydroxytryptophan. If you care, this guy is usually 5-HTP, 5-hydroxytryptophan, whereas this guy is 5-HT, 5-hydroxytryptamine. But 5-HT is AKA serotonin. Serotonin is one of your neurotransmitters. Serotonin is acetylated to make N-acetylserotonin, which means we involve an acetylation step. And then N-acetylserotonin is SAMmed to melatonin. What type of reaction is that? One carbon transfer, fantastic. N-acetylserotonin is one carbon transferred to melatonin, which is made in the pineal gland, involved in the sleep-wake cycle, and involves SAM as the transfer. So remember, just so I'm reminding you guys, anytime you see SAM, what amino acid is involved in the pathway? Methionine. So just in case he asks an oddball question where he talks about what amino acid is involved in serotonin synthesis, the primary amino acid is tryptophan. But he could also throw, have a good night, no worries, he could also throw methionine on there because it is involved in the synthesis pathway. So 5-hydroxytryptophan, 5-HTP, is turned into serotonin, which is decarboxylation by PLP, forms 5-hydroxytryptamine, aka serotonin, which is acetylated, which is methylated to form melatonin. So tryptophan makes the nicotinamide portion of NADP. It makes serotonin and it makes melatonin. If you need a little mnemonic, the tryptophans makes theonins. Tryptophans makes theonins. Just memorize that so you don't get confused between tyrosine and tryptophan on your test. Now, he kind of talked about this in class. Did he talk about the breakdown of serotonin with you all? Okay. This bad boy breaks down serotonin. It also breaks down your catecholamines. You're going to talk about that in one of your other courses, or you did with Dr. G in Try 1. Serotonin is broken down by monoamine oxidase. Monoamine oxidase. It just simply oxidizes monoamine. And this is a monoamine. It's a tryptamine. So monoamine oxidase breaks it down into 5-hydroxyindole acetaldehyde, and that gets broken down even more. The reason you know this is a breakdown product and the way you can remember it is indole. Where did you see indole otherwise? You guys recognize that word? Microbiology. When you did the breakdown of tryptophan and you looked for tryptophan breaking down amino acids and you had Kovacs reagent in microbiology, you were looking for the breakdown of tryptophan by testing for the indole ring. Because when tryptophan breaks down, it breaks down into an indole ring. When you throw Kovacs reagent in there, it turns red. Right? It makes that red ring, which indicates the tryptophan's been broken down because you had an enzyme that was tryptophanase. So, monoamine oxidase is a tryptophanase, or serotoninase. It also breaks down your catecholamines, all your monoamines, and it turns it into an indole ring, basically, and then breaks it down further from there. All right. Says that all on that page. We're not going to go over it again. I kind of simplified there. Like I said, tryptophan makes your onin. Serotonin, melatonin, and nicotinamide portion of NAD and NADP. This is the last page. There's 3 quick reactions on here. It's not going to take me a little while, and I'm not starting chapter 10. Chapter 10 tonight. I'm going to fix that. We're having some technical difficulties over here. Please stand by. Okay. It's fixed. I'll get some duct tape. Cool. So, any questions on tryptophan before I move on? Last couple reactions. You guys have been troopers. Today's a long day. Sorry. Glutamate serves as the precursor for the inhibitory neurotransmitter GABA-aminobutyric acid. Please, please, please know that statement. Glutamate serves as the precursor for the inhibitory neurotransmitter gamma-aminobutyric acid. So if he asks you on the test, I'll get to it at the end of this. It's a TQ that got me. Glutamate serves as the precursor for the inhibitory neurotransmitter GABA-aminobutyric acid, gamma-aminobutyric acid. The enzyme that does this is glutamate acid decarboxylase. Glutamic acid decarboxylase. Glutamic acid decarboxylase or glutamate decarboxylase. What cofactor does that require? Decarboxylation. PLP, aka B6. What else does B6 do? Yes, transamination. You guys have learned something. One less thing we have to do. You guys have learned a lot. One less thing we have to do in chapter 11, though. So, glutamic acid decarboxylation requires pyridoxal vitamin B6. It's a decarboxylation reaction. B6 also does transamination. Glutamate is converted to gamma-aminobutyric acid. It's a one-step process. So if he were to ask you on this theoretical test, if I were asking a question, it might be something along the lines of, What type of reaction is the one-step conversion of an amino acid into an inhibitory neurotransmitter? What type of reaction is the conversion? What type of reaction is the one-step conversion of an amino acid into an inhibitory neurotransmitter? That is a very deep question. You got to know what we're talking about when we say inhibitory neurotransmitter. You got to know that we're referencing the reaction. The answer to that question is decarboxylation. Decarboxylation is the type of reaction that is the one-step conversion of an amino acid into a inhibitory neurotransmitter. You guys get that? Cool. Okay, we made GABA. Oh, by the way, these are all on your sheet somewhere. I think on the front page, right down here. Last three are going to be right here. Bottom right corner, front page. Histidine is the precursor for histamine. That makes it really easy. If he asks you what makes histamine, pick histidine. Histamine is released from inflamed, I didn't know you could inflame it cells. Histamine is released from inflamed cells or injured cells. It's responsible for bronchoconstriction and vasodilation. How does it bronchoconstrict and vasodilate? You're going to hate me. All these crappy old questions. Like, kind of. Not really. It's the opposite effect of aspirin. How does it inflame? What does histamine do? Histamine activates phospholipase A2, which frees arachidonic acid from the cell membrane, which gets converted by COX to prostaglandins and thromboxanes, which cause bronchoconstriction and vasodilation, and leukotrienes cause bronchoconstriction. So histamine activates the eicosanoid pathways. Histidine activates phospholipase A2. Way old stuff. Probably won't come up on your test. Maybe on the final. Histidine is decarboxylated to form histamine by histidine decarboxylase. What does this? Pyridoxal phosphate. Good. Histidine to histamine decarboxylation. Bam. That's all you got to know. And lastly, arginine produces nitric oxide. Have you guys talked about nitric oxide in physio yet? Okay, nitric oxide is one of the most important vasodilators in the human system. Very important vasodilator, produces lots of local vasodilation. It's catalyzed by nitric oxide synthase, makes the enzyme really easy to remember. What makes nitric oxide? Nitric oxide synthase. What you do need to remember, though, is it comes from arginine. And you do also need to know it's produced by the vascular endothelium cells, and it's a vasodilator. What that means is that when you need to vasodilate, the endothelium, the inside of your blood vessels, produces nitric oxide and causes local vasodilation. Nitric oxide, which is produced from the precursor arginine, is produced by nitric oxide synthase. It's produced by the vascular endothelium cells as a local vasodilator. So we produce nitric oxide. It goes out to the smooth muscle cell. What smooth muscle cell are we talking about? Smooth muscle cell in the blood vessel. Good. That's what you were saying. I heard you. Smooth muscle cell in the blood vessel. It acts through the guanylate cyclase, which is a type of G protein coupled receptor, and it produces smooth muscle relaxation, which allows for vasodilation. So on your sheet in the bottom, this is the only one I'm going to talk about. The other two are pretty easy. Histamine, histidine, inflammation, injury, bronchoconstriction, vasodilation. But down here, arginine produces nitric oxide by nitric oxide synthase. Nitric oxide activates guanylate cyclase, which is a type of G protein coupled receptor, produces cGMP from GTP, just like we produce cAMP from ATP, and we get smooth muscle relaxation and vasodilation. Smooth muscle relaxation causes vasodilation. That's it. That chapter is a beast. It's like 8 pathways. Remember. Ask or know which amino acids are involved in which pathways. Know your first nucleotides, your order of synthesis, your rate-limiting enzymes, and stuff like that. Before you guys leave, I want to point out real quick, if you want to get started or if Dr. Sarkar hits this before I do, this little Biochem 2 vitamin chart that is on the website and that I showed you earlier is my cheat sheet for chapters 11 and 12. It's all your water-soluble and fat-soluble vitamins. It's broken down by vitamin, precursor, enzyme, the active form, the function, the recommended daily allowance, which I'm pretty sure he tells you to ignore. So if you want that, great. If you don't, ignore it. and what deficiency states cause. So if you want to get started studying, or if he hits it before I do in tutoring, you are welcome to do that. But we will be talking about that next Monday, along with Chapter 10, which will take like 15 minutes. So thank you guys for being here, you troopers, and for laughing at my crappy jokes. And for laughing at me cussing and insulting and saying inappropriate things on a vodcast once again. You guys have a good day. Yeah. And I will see you all on Thursday with delicious food. If you're listening to my vodcast, you better bring something delicious for us to eat, because we are having a joint tri-luncheon, if you guys didn't know that. Delicious food.